Good morning, everyone. My name is Chad Whitman. I am the preclinical sales manager here at Synthego. And so today I'm going to just present rapid CRISPR results from discovery to the clinic and for a variety of different products. So to start, what I want to show is some data to illustrate the rise of CRISPR and how it has taken over helping people with different therapies. So if you look at the entire world, the United States, well, the main three components of the world, the United States, Europe, as well as in APAC, there have been over 1,000 therapeutic developers out there with almost 1,000 trials that have been conducted around cell and gene therapies. And a lot of these are even sponsored by academics and the governments of these countries where this is occurring. And there's a huge amount of money, $6.7 billion that's been invested. So while CRISPR itself has come into prominence a little over 10 years ago, there's a huge promise of what this can do to help treat a lot of different diseases. An interesting statistic to look at is that the use of CRISPR in terms of new gene editing trials has been increasing year over year and has become more prominent than other methods out there that have been historically used for gene therapies, so to speak. So today we would say that 70% of all new gene edited trials are CRISPR based. And currently, especially in the United States, there are 4 CRISPR-based trials that are already in our phase 2 and 3 levels that could be hitting the market very soon, including one most recently that got an approval that will likely get approval in the next month or so for sickle cell. So to start, what I want to show you is 3 numbers that kind of illustrate the challenges that people have when they do CRISPR, putting them into a therapy. And this was data that we collected from our customers, as well as our customers out there, what were their struggles. So the 45% of people said that one of the hardest things to do was to actually get a timely procurement of the reagents. 47% of people told us that it wasn't clear of what the regulations were going to be. And another 26% of those people also said that there was challenges when they did discovery level experiments and they later got to a clinical. So what we want to help people do is reduce these issues. And for example, help them get more accurate timelines to receive their products, help them better understand the regulations that they're going to have to go through to get their therapy approved, and also help them become a single vendor that can walk them through the beginning experiments all the way down through the clinical challenge. And so by doing this, Synthego wants to do is help people get through these challenges and have a more successful and faster way to the treatment. So I started with that background to kind of give you the bigger picture of what Synthego is looking to do. What I'm going to present now is more specifics about our products and how they kind of fit your solutions in a lot of different ways. So the main thing that we focus on, or that my team focuses on, and I work a lot with Korea, is on our synthetic sgRNA. So with our synthetic sgRNA, this can be people using this in a discovery setting where it's more like a research, testing out a lot of different guides, to something that is preclinical. So before you're getting to the clinic, but maybe now you're testing in animals, and then downstream to the clinic where you need a higher quality and a more well-documented product for the regulatory. So at Synthego, we split these up into 3 kind of products. The main one that I'll focus on is what we call RUO sgRNA. But I will also mention a little bit about what we call IND enabling, which is more of a reference to some of the American approvals that you do with the FDA here, but then also GMP, which is the Good Manufacturing Practice, which is very regulated and documented. Okay. The main product that we deal with a lot of researchers is what we call synthetic sgRNA. So this is chemically synthesized guide RNA. So rather than expressing this from a plasmid, we are actually synthesizing RNA molecules one base at a time. So most of what people have been focusing on for this is sgRNA, or I will say guide RNA, that's a term I will use a lot here, but guide RNA that is for SP-Cas9, which is the most common CRISPR nuclease that is used in the market. One of the things, though, is that we actually can synthesize guide RNA for other nucleases. And so I'll talk a bit about that in a sec. But so when we say synthetic sgRNA, this is the chemically modified guide RNA that we synthesize one base at a time. So what has been shown over many years now is that you can actually synthesize these 100 mers, or these, especially for Cas9, in this method, and they work extremely well compared to other two-part products that are out there. So what we see on the right over here was a comparison that was done many years ago, comparing our guides versus another competitor's guides. But the bigger thing I would point out is that the guides, this part here was they actually had two different RNA molecules they purchased and then synthesized together, versus these other examples here on the left that were a single molecule. What we demonstrated with our product was that we could get much higher editing efficiency in the cells with our single guide that was made. So now this has become a very common solution that researchers come to us for. In terms of, what can you use this for? This stuff is used for knock-in as well as knockout experiments. And I'll show a slide a little bit later that kind of illustrates this from a let's say a biological level, so you can understand it in case you don't. Now, what Synthego offers is two different products that fit this research use only, or RUO. The main one I mentioned, we call it synthetic sgRNA, and it's where you give us a 17 to 23 nucleotide sequence that targets specifically where you want to make your edit. And you can design this with software. And then we synthesize that onto an 80 mer or 80 base RNA base pair strand that we start with. And then that's what we send to you. If you are someone who uses a different CRISPR enzyme to do, or a different Cas enzyme to do CRISPR editing, that is under what we call our advanced RNA portfolio. And so what I've listed here are three of the most common ones that we come across: Cas12a, also known as CPF1, SA-Cas9, as well as Cas13a. But there is a large amount of nucleases that have been found out there that you can explore for different applications. In general, we say that between 20 and 100 nucleotides is what we can synthesize with RNA, but we have the ability to go a little bit larger than that, but it takes some additional review on our part. So I would always recommend, if you're ever interested in Synthego providing you with guide RNA, it is to actually engage with Philly Korea of what your project is, and then they will work with me to see what we can do and how we can support you. That product is focused on the standard research applications that people may do. We have the ability to increase or offer additional characterization that is necessary for government, that is for submission of a gene therapy for a therapy, for example. And so this is where we will do additional testing on purity, as well as the sequence itself, and then proper documentation. And then when we get to clinical guides, then we actually have some additional standards that we are working with in the US, the FDA, on what are the standards that we should be having in these products when they go to therapy. So in terms of what we've done, we've been very successful helping people. That's what this slide is telling you, that if we work with a lot of different companies, we've helped them get through the process. Now, what's really cool is when you look at the broad spectrum of what has Synthego done, we have synthesized over 1 million guides during our time. We've been cited in at least 1,000 publications and have been helping researchers all across the world, including those in Korea. So that's kind of the overview of guide RNA and kind of carrying this into bigger applications downstream. The next thing I'm going to present is talking about scaling experiments to where you can look at a lot of different targets. So what I focused on before was more of you need to do a CRISPR experiment and you have a guide RNA that you need synthesized. That's how I started this. What I'll present on now is saying, let's scale that specifically when you're trying to identify targets and knocking them out. So starting out, what I'm going to walk through is the basics of a simple versus making this very broad. So this is where I will end up talking into our libraries that we offer. So let me back up a bit in terms of some standard terms that I may use to help make sure everybody is on the same page and understands what I'm going to present. The way that we look at doing CRISPR is creating what we call the ribonucleoprotein complex, or RNP. And that complex is a combination of a specific SG or single guide RNA that is actually mixed with the nuclease, in this case Cas9, and pre-mixed. That then is fed into the cells to do the CRISPR experiment shown here, and the editing shown here, starting out with the scissors. Now, the way that CRISPR works is that it causes a double-strand. The way that the general way I'm referring to CRISPR here creates a double-stranded break. Now, there are two different paths to which the DNA can repair itself. One is the process of non-homologous end-joining, where the cell will actually try to repair the genome, but may cause an insertion, a deletion, a frame shift. But it's going to just do it the quickest way possible, depending on what's present there. This is the most standard way that eukaryotic organisms repair when they have double-stranded breaks. Another type of repair that I will highlight at the end briefly with another line of products is our homology-directed repair, where you actually make the cut and then you bring in a DNA template to actually change the sequence that's there. And the term knock-in is synonymous with changing a single nucleotide variant or a single nucleotide polymorphism, so SNV or SNP. It's also utilized to delete a sequence as well as even insert a sequence. And what I'm mentioning here is a tag, or for simplicity, something like GFP or HA, different tags that you might put onto the N termini or a C termini of a protein. So I mentioned that we focus around the ribonucleoprotein complex, or RNP. And the reason we do that is we have seen that this is a better alternative than to using plasmids. So let me show you some data to kind of explain why our reasoning behind this. So on the left, what we're showing you is that we are this data is showing you the indel frequency on the y-axis, meaning how efficient are you at creating an in-frame deletion into the sequence, which is the measure of success of this particular editing experiment. So what we're showing you is that as you increase, even when you increase the amount of plasma that you're adding, shown here in the different shades of black to dark, almost black to light gray, for various targets, you do get increases in efficiency. However, what you notice is that the two bars here in this, let's say teal and green, where we're using the ribonucleoprotein complex in different ratios, we get significantly higher editing efficiencies. So it's telling you that while you can increase the amount of plasmid that you're putting in the cell, you're still not getting the same efficiency of creating the intended edit that you can by using an RMP complex. There's also another positive to this, because one of the challenges is that when you increase the amount of plasmid, you actually can hurt the cells, because now you're causing other problems within the cell by bringing an exogenous DNA in there. So what the data shows you is that as you increase the plasmid, what decreases is this luminescence result that you see on the y-axis. Well, this is measuring the viability of the cells after they've been transfected. So what's noticeable is when you increase the amount of plasmid, you may get more editing, but you're also hurting the cells. As you notice, the cells become less viable with more plasmid. However, the ribonucleoprotein is very stable in terms of the viability of the cells, regardless of the ratio. So it tells you that you can have more confidence using this product to give you a better edit, as well as keep your cells more viable. So taking this approach, one of the things that Synthego wanted to offer was to help people get better knockouts, to where they could trust the chemically modified guides that we had created, but then also be able to have a very effective way to create a knockout for a particular gene in their cells. So what we created is a product, is something we call multi-guide. And what multi-guide design does is we focus on, usually within the first exon of a human or a mouse gene, what we are doing is designing 3 different guides to basically create that indel that I showed earlier, that deletion that causes a frame shift within this gene. You can definitely do this with a single guide. And I would say, if you were ever creating a therapy where your therapy was to actually bring in a guide to create a knockout of a particular gene, you would want to use one guide. But if you're trying to evaluate whether a gene, the presence of a gene affects your drug treatment or any kind of other experiment, we actually recommend using this method, because what we see is that when you look at a single guide versus 3 guides, you get a much greater ratio of indel created versus the broad range that you could get with just a single guide. And the indel that you create actually makes a knockout. So what does that mean when I say it creates a knockout? So if you've ever looked at Synthego's ICE or ICE software, what we do is actually have you input in Sanger sequencing files. And in there, we look at the spectrum and evaluate of the sequence and where you created the edit, what types of deletions have you created that may have caused frame shifts. Because the point with this product is you actually change the DNA sequence to where you create a stop codon shortly after that edit that then does not make a functional protein. And so when you look at our sequence, what it's looking for is, look at your sequence in our software, it's looking for how of the sample you sequenced, how much of that sample has a change in the sequence. So the protein that gets made from this DNA sequence is not the full length protein, hence it is knocked out. So that's what our software does. And following the seminar, if there's any questions about how this software works, we can provide, or I can provide some links to share, to read and see how we do this, as well as the website to test this out. This is a free, the website that's evaluating this is free. Anybody can use it. There is no charge to use it. So if you create a knockout in a different, with a different method, you can always use this software. There is no charge to use it, and it'll tell you how successful you were. So I'm a scientist at heart. I like to see data that shows me how good is the multi-guide approach to individual guide editing. So this is a simple example that we did with Dr. Jonathan Weissman, who previously was at University of California, San Francisco, has now moved out east into the Boston area. But while he was at UCSF, he was one of our earlier customers. And what they did with our technology is they got our multi-guide knockout kit and compared its editing of the 3 guides together versus those guides by themselves. And so what the data shows you is that if you look at each of the individual guides, you've got a approximately 40 to 50% in this TNF alpha, for example, where you created a deletion, whether it was a small amount was greater than 50 nucleotides, some that were less than 50 nucleotides, and there was some alteration to the original sequence, versus when you use the multi-guide approach for that gene, your editing efficiency was almost 80% for this example, and say 80% and 80% for these other 2. So there was a significant difference in the editing ability of using the 3 guides. What's also really powerful about data like this is he did this in primary cells. So not a standard immortalized cell line like HEC293 or K562, like cells that I had used in the past. This was actually primary cells, which are much harder to work with. So that's one of the values of this technology, is it works very well with immortalized, with iPSCs, and even dendritic cells. So now what I've shown you so far is a single example, but one of the things that we can do very easily at Synthego is you can come to us with a list of targets that you want to explore. And what we can do is we can actually plate those multi-guides into individual wells on a 96 well plate, to where you could, or a 384 well plate, to where you can actually explore a lot of targets. So a good example for this is, let's say that you are trying to do a screen and identify in your model system which genes are important for a certain treatment. So what this approach, this arrayed approach, enables you to do is you could choose a whole host of genes that you want to knock out. You give that list to Philly Korea, who works with us. We will create this, we call it an on-demand library for you, where we will plate it how you want it. And then you would receive a plate where you would then mix the wells with Cas9 protein that you can purchase. And then you would then transfect them into cells and do your experiments. What's valuable about these types of projects is every well is one gene. So it's not a pooled approach where all the targets are mixed together and you later have to clean up the data to find out which targets, what targets contributed to what. In the arrayed approach, the value is whatever you look at in that well is the effect of one gene being knocked out. So show you another example of the power of this that the same lab at UCSF did. Again, another set of primary cells, and what they did is they actually looked at a large number of genes to see how effective or try to find out, trying to knock out a lot of different genes. What's very, if you notice, if it was very effective at knocking the gene out, you see very little gray. But what this is telling you is sometimes it doesn't always work. And that is one reality of working in biology, is we may try to knock out a gene, and the cells will prevent us from knocking it out. So the nice thing about this data is this allowed them to evaluate their system and find out which genes could be knocked out very easily in these cells. So there were certain genes that weren't, some that were easily knocked out, and this gave them some very valuable information. Again, it was also in a primary cell line, which are much harder to work with. And our guides have been very well demonstrated to work in all different types of cells. So another thing that I'll briefly talk about is Synthego's capability to create the edit for you. So Synthego has a platform we call Eclipse, where we can do is create edits for you. And what we've done is created this to be very automated. So there are very few human interactions to actually create the edit, which enables us to make these more consistent and faster than trying to have somebody do all this themselves. So what you see in the picture here on the right is a screenshot, or is a picture from our factory of one of our work cells. And this is where cells would be brought in after the transfection, cultured, and evaluated. So this is part of the process of what we do. We've done this we test this, every cell line that's come through has been evaluated for 200 different transfection conditions. We have done this this is actually out of date, with over 800 cell lines, and created over 250,000 edits. And what you can see here is the different tissue types that we've evaluated over the years. To give you more specifics, these are some of the most common cell lines that we've worked with. And what I've put in here is the name of the cell line as well as the tissue, and also have noted iPSC. Now, one thing that we are able to do, and this is something that we do in our consultations that Philly Korea could tell you more about, but if we can provide the cell line ourselves, then we get it in-house, we do the edit for you there. But if you needed a particular cell line of yours edited by us, we can accept cells from the customer. So understand that there is a lot more involved with shipping cells from Korea into the United States, but it is a capability, and we do a lot of work in iPSCs for customers. So the type of edits we do are the knockouts and the knock-ins, such as tags and SNVs or SNPs, the nucleotide variants. And this is some data to kind of show a lot of popular cell lines that are immortalized cell lines, and us doing basic knockout experiments, but also in iPSCs, us doing a lot of different tags. And the general thing I would say is that it's something that we're very good at, and we do a lot of these projects. So with that, I was going to stop. So I am happy to answer any questions. Thank you.